Good afternoon, everyone. My name is Jing Gong, the host of today's webinar, and also the tech support working at Biological Europe. I would like to welcome all of the attendees and specifically our speaker to Biological webinar today. Before I introduce today's speaker, I would like to quickly remind all the attendees that it works best if you enter your questions into the Q&A channel. Also, it's possible that you can enter your questions in the chat channel. At the end of the talk, I will verbally walk through all the questions and get as many of those answered by our speakers as we can. So now it's my pleasure to introduce you our esteemed speaker, Dr. Alex Dulovic. Since beginning of 2022, Dr. Dulovic is a deputy group leader of the Multiplex Immunoassays Group at the NMI in Tübingen in Germany. He has received his master's degree in industrial and commercial biotechnology at Newcastle University in UK, and later his PhD at Max Planck Institute for Biology, Tübingen, under the supervision of Professor Adrian Steit. Following a short postdoc at the MPI for Biology, he joined the NMI as postdoc and project manager in 2020, and later becoming the project leader. His group focuses on development, validation, and screening of multiplex serological immunoassays for the detection of pathogen infections, as well as implementation of multiplex serological immunoassays in epidemiological cohorts. So without further ado, I think it's time that we can hear the talk from Dr. Dulovic. So Dr. Dulovic, I think you can start share the screen now.
Yes, thank you very much for the introduction. I hope that you can now see my screen and also all hear me. This will be good. So yeah, I will talk today about various work that is done within our group. So we are from the NMI, Natural Medical Sciences Institute. We are an affiliated institute to the University of Tübingen. And my name is Alex Dulovic. I'm deputy group leader here of our Multiplex Immunoassay Group. And I will run through today some of the work that we do on multiplex immunoassay development. And specifically how we use this to assess antigen-specific antibody responses. So I will start very briefly by I think Jing's already given a very nice overview of me. So as you said, I'm originally from the UK, where I got my bachelor's and master's degree, and then completed my PhD at the Max Planck Institute for Biology, working on parasitic nematodes. After this, I then joined the NMI, first as a postdoc and project manager within the then biochemistry group, and now since this year I've been a project leader and deputy group leader of the Multiplex Immunoassay Group, which is one of the two groups that has evolved out of this biochemistry group. The NMI itself, so we are an independent institute with a focus on application-oriented research at the interface of life sciences and material sciences. We are affiliated to the University of Tübingen. And we've been established now for nearly 40 years. We are a nonprofit foundation with currently around 200 full-time employees engaging in research across multiple fields. And we are also a member of the InvestBV group, along with similar sort of types of institutions and also commercial companies within our area. The institute itself really focuses on three main fields: pharma biotech, for which we are part of on the left here, as well as then other specialized areas looking really at medical engineering and also on material sciences. And within the pharma biotech area, we are one of several groups working really on biomarker identification and analysis. And explicitly also, there are other groups then who work similarly with multiplex immunoassays or on drug development support or development of cellular testing systems. But for us, our real expertise is within immunoassays itself. So this is a very typical workflow that we would use. So we are involved really at all stages of the process. We are not limited to just developing biomarkers. This can also be development of specific reagents or characterizing their performance. But we are also a measurement facility. So we take part in large numbers of studies ranging from really very small amounts with tens of samples, all the way through to very large epidemiological cohorts with 50,000 plus samples. In terms of the work we do, every single assay that we develop or every single piece of work, whether it's a bead coupling, we always validate its performance. And traditionally for assays, we will do this using a fit for purpose approach according to FDA or EMEA guidelines. But we are also very flexible in terms of the types of samples that we analyze. It is not just serum or plasma or typical blood products that we would work with. We also work often with saliva samples or with CSF samples or with samples from tissue lysates. And for each one, we always then validate it so that we can ensure that we are receiving exactly the same level of performance, regardless of the sample matrix used. We then obviously always evaluate our data and report it onto the customer, although now this is obviously much more common in the form of publications. What our particular interest is, and something that we work very strongly within the group, is on the development of multiplex-based bead-based assays. For anyone who is unfamiliar with this technology or these techniques, this largely uses principles from the Luminex company, and specifically the use of magnetic bead IDs. For this, I will very briefly introduce this. So the magnetic beads themselves effectively can be separated into spectrally distinct populations on the basis of combining several different dyes, which is shown here by the letter A. So you have a combination of an orange dye, a red dye, and an infrared dye at different concentrations, which allows you to generate a specific emission spectra that, when excited, allows differentiation between different populations. And this obviously allows you then to have really an unlimited number of IDs simultaneously. The current maximum you can work with, just due to the limitations of those beads that you're commercially available, is 500, although there are groups that develop their own beads with over 3,000 different IDs. And as this is a bead-based format, it actually very nicely leads itself to being automated in a lot of processes. So with us, at least, we use Biomech i7 pipetting robots as well as Kingfisher machines to automatically couple our beads. And the very basic principle which guides all of our, well, the vast majority of all of our assays is shown down in the bottom right corner. And this is that for each of these magnetic beads, we couple an antigen from whichever pathogen or parasite or cytokine, whatever our target of interest is. And then when we incubate this with the sample, in this case shown here is an example of just a rat IgG antibody, we then can effectively capture antibodies that bind to this antigen, detect this binding through the use of a PE-labeled secondary antibody. And when we then run this through the machine, we have 2 laser lines in the standard FlexMap 3D or 3 laser lines in the modern IntelliFlex system that allow us to firstly differentiate the different beads and separate them out into populations for our different antigens. And then secondly, to actually measure how many antibodies are physically binding to this bead. And particularly of the new development of the IntelliFlex of having 3 laser lines, this means that you can measure multiple Ig types simultaneously, such as IgG and IgM. Now, within our group, we work on a wide variety of different pathogens and have a large number of diseases of interest. In this talk, however, I'm really going to focus in on these 5, which is SARS-CoV-2, Borrelia or Lyme disease, as it's more commonly known, the 2 respiratory viral pathogens, RSV and Influenza, and then also some work that we do on emerging pathogens, which explicitly now is for Monkeypox. I'll finish my introduction by just giving the very basic overview of our principles of assay development. So every single assay that we develop always has exactly the same principle behind it, that these should be reproducible, low threshold assays to study diseases of interest or mechanisms of interest. Where possible, we like to be able to differentiate between vaccinated and infection eliciting responses. Where appropriate, we would also like to be able to investigate function as well as presence of the antibodies themselves. And if there is cross-reactivity or cross-protection from or towards closely related species or between variants of the same pathogen, they should be able to be investigated with our assays. So I will begin now with SARS-CoV-2 and some of the work that we have done on this. So I think everyone is fairly familiar with this and probably quite fed up of now having to hear constant talks about SARS-CoV-2. So we'll try and keep the introduction to this as brief as possible, but obviously since the initial outbreak, we have now developed into a global pandemic. And we have had vaccine development and deployment pretty much throughout 2020, with then these schemes becoming globally quite well established throughout 2021 and 2022. However, simultaneously, we have had the continuous viral evolution into variants and subvariants. So, for example, all of our work in 2021 was really focusing on, well, the initial vaccine responses of 2 doses, combined with also differences towards the alpha, beta, gamma, delta, and very beginning of the BA1 variant of Omicron. But obviously now this year, we've since had separate waves of BA2 and now BA5. And over the course of pandemic, there have been a wide variety of assays that have been built commercially and academically. I think almost all groups who are involved in this have developed something of their own. And within our group, we developed 2 assays. The first one of these, Multicov AB, and the second one, RBD-Cov ACE2. Multicov AB is a simple antibody binding assay that looks at several targets of SARS-CoV-2. Instead of classical ELISAs, such as those from Roche or Euroimmune or something like that, that used single analytes, we had always the full trimeric spike, the RBD S1, S2 domains of the spike, plus the nucleocapsid, to enable us to really differentiate between the different antibodies produced towards these antigens. Our second assay, RBD-Cov ACE2, this uses the same principle of our bead-based technology, coupled in this case with different RBD variants. And when we incubate then sample material, so which has neutralizing antibodies present, in the presence also of biotinylated ACE2, what we can do is effectively have a competition between whether the biotinylated ACE2 or whether the neutralizing antibody can block to and bind to these RBDs. And therefore, through this competition, when we then add a secondary antibody, we can actually measure how successful were these antibodies in blocking this interaction. And this is effectively a classic sort of pseudo-neutralization assay. I will come very briefly onto the actual details where we could show that the performance was the same, but obviously had the key advantage of allowing us to look at an unlimited real number of variants simultaneously. So in terms of our Multicov assay, this, in terms of the very basic performance, so we use here, unlike these classic ones from say Roche, Siemens or Euroimmune, we use a dual antigen detection system, the spike and the RBD, in order to determine positivity. And what we could see here was that this actually gave us then the highest sensitivity and specificity of all of these assays that we tested. So by using multiple antigens, we could already actually improve assay performance before we started looking at these differences within a population. For the RBD-Cov ACE2, as I said, our performance of the assay itself is actually very similar to a classic BNT. So you can see here the correlation between the titer, which is produced from a typical assay, versus the percentage inhibition that is produced in our assay, and the nice correlation between the two. But as this was multiplex, it means that we could not just measure the wild type response, but we could look at any number of variants or any number of mutants at the same time. As the pandemic evolved, we could obviously then continually increase this assay and look at new responding variants. And the key other part of this assay was that it was validated and optimized not to work only with human samples, but also with mouse and rat, which means that it was actually suitable to be used for both preclinical and clinical vaccine development, and that you did not have to undergo changes in assay or anything. You could really just use it straight away after we'd obviously done the initial work, and switch between species as your projects evolved. So very briefly, at the very beginning of the pandemic, we already actually started to gain some very strong insights into how our setup of using these multiple analytes to assess different antigen antibody responses could be effective. And this here was some work at the very beginning of 2021, where we could see that due to having multiple antigens, in this case, not just the RBD or spike variants, but also the nucleocapsid, we could actually differentiate between individuals that were infected and individuals that had been vaccinated. So the individuals that are infected, you can see here are red circles. They largely cluster towards the top right of the graph, whereas individuals who had been vaccinated, these are shown in blue. And whilst the spike responses are almost identical between the pair of them, there was actually a massive difference then in the nucleocapsid. As expected, obviously, if you were vaccinated, you did not really produce the nucleocapsid antibodies. And this means that even if we didn't know what a sample status was, we could straight away differentiate whether it came from an infected person or from a vaccinated person, just by using multiple analytes. Now, as I said, this automated format, and because we are bead-based, it means that we could semi-automate all of our assay setups. So both the multi-cov assay and the RBD-cov assay could be automated, and this means that we could actually measure in excess of 700 samples daily with them. And so this here is really data from a sub-cohort of individuals of a German-wide seroprevalence survey called MUSPAD, where we were really interested at the time in assessing differences in the performance generated by different vaccinations and different vaccination schemes within the general population. And you can see here that, for example, people who had received 2 doses of an mRNA vaccine, this MM or PP for Moderna or Pfizer, had these much higher, significantly higher responses than the individuals who'd received 2 doses of AstraZeneca or AA. And when we then compared this to people who had then received this kind of mixed assessment of a first dose of AstraZeneca and then a second dose of Moderna or Pfizer, we could see that this then actually boosted the responses significantly and actually brought them up much more similarly to people who'd received 2 doses of mRNA in the first place. And the nice thing about this is you can see with the ends here, these were large sample cohorts that we could look at. So unlike other studies where you are obviously limited to a certain degree in the amount of samples that you could look at with the assay type, here we could evaluate in excess of 700 samples, for example, of people who had received 2 doses of Pfizer. And another key aspect of this that came in that really showed us nice results that we could gain from using our multiple analyte format was that we could then compare actually not just the amount of antibodies people had towards different analytes, which you can see here on the left in the heat map, but we could then actually compare how these ratios differed between the different vaccination schemes. So for example, on the left here, you have a heat map showing titer, described as a Z-score, for all of for the spike, RBD, S1 and S2 antibodies across the different vaccination schemes. And the key part I want to draw your attention to here is that people on the left in this AA group for 2 doses of Astra or this single J for the 1-dose Janssen vaccine, they are largely gray for the spike RBD and S1, indicating that they were at the very lowest end of titers that we saw within the assay. By comparison, the people who have received the MM or PP, well, they are largely red across these same ones for the spike RBD and S1, indicating they were among the highest performing. And it's instead they have for their S2 more samples in the gray. And when we really then look at this for this comparison between the S1 and S2, we can see that the 2-dose mRNA-based vaccine really produced around 100 times more S1 antibodies than S2 antibodies, whereas this 2-dose Astra vaccine or the single-dose Janssen, it was only around 30. So you had a 3-fold difference in actually the amount of S1 directed antibodies that you were producing through receiving this mRNA vaccine. And this was also then seen by several other groups using different formats. And we suspected, as what they did, that this was a potential reason for differences in efficacy between the vaccines, as obviously these S1 directed were more functional and were more neutralizing than these S2 ones, which were just tended to bind the virus. And I would bring now in, it's not just vaccinated samples that we can look at, and this can gain insights from this format, it's also infected samples. So here this was a large cohort of adults and children who had been infected from the same households during the first wave of the pandemic. And we can see here that really depending on the analyte that you look at, you will see significant differences between your sample cohorts. So for the spike, for the RBD and the S1, you can obviously see there are very significant differences with children having much higher antibody titers against them than adults do. However, the same is not true for the S2 domain. For the S2 domain, there is broadly speaking no difference between children and adults. And this is not a finding that you would gain if you'd only evaluated this with a single analyte type of assay, such as the RBD. And when we then also looked at the nucleocapsid, we could see here, yes, there actually was a strong difference. There was a significant difference between them, but this was much reduced compared to, say, the RBD or some of these other analytes. So we gained information here not only on differences of the sort of titers for the antibody and antigen specific responses within our population, but we could also gain the differences here between the different antibodies and antigens themselves. Now, obviously, as the pandemic then evolved, variants of concern and these continual mutations started to play larger and larger roles. And so here, having this bead-based format and being able to always incorporate new analytes and new variants into it meant that this was actually a very useful tool for us to be able to actually study differences and how these new variants would be able to evade or respond towards current antibodies, either generated through infection or vaccination. So here, for example, this was work that was done through the middle of 2021 up until really the emergence of the Delta and the Kappa variants. And what we could do was, by using our assay and by putting in all of these different variants that were circulating or had been circulating previously, we could really then assess and identify the presence of individual mutations in the amino acid and how they were actually strongly affecting these differences in responses. So here, for example, you can see that when you get to the very bottom end of the scale, these sort of eta, gamma, theta, and beta variants, these all possess this E484K mutation. And when you actually then had this N501Y in addition to this, as is seen for gamma, theta, and beta, this effect became even stronger. So obviously, we were not the only group to see this. Many groups identified these critical mutations, but what we could see with our assays is that we could do this all simultaneously using a fraction of the amount of serum. This entire work requires 5 microliters of serum. And we could then really enable ourselves to actually identify variant-specific differences and how these variants changed their role and how they affected the actual inhibition that could be generated. And obviously, since this work was done, we have continuously always kept on developing our assays to incorporate new variants. So coming up to then the end of last year and beginning of this year, we then had similar work looking here at now with the BA1 and BA2 variants of Omicron. And we could see fairly easily that these obviously effectively evaded the neutralizing antibodies that were already present from vaccinated or infected people, both of them. And that we could also compare this and see, was this similar, for example, to beta, or was this similar to other variants that we could gain an idea of how this was likely going to play out. And obviously, since publication now, this has continued. So we now have also BA 1.1, BA4, BA5, 2.12, all available and within the assay already having undergone validation, and 2.75 is within progress. And by and large, from the time that a new variant does emerge, our largest waiting time is physically for production. As soon as the antigen is actually available and it's ready and can be incorporated within the assay, we can effectively have it in, have validated its performance, and be able to already utilize it and gain useful results in a matter of weeks. The last point I'd really like to talk about with regards to these SARS-CoV-2 assays is just that the sensitivity of both of them actually enabled us then to really screen and look at individual responses within vulnerable cohorts, because while there is obviously advantages in being able to look at population-wide changes, being able to actually investigate individual specific responses, particularly within those most vulnerable, has large implications, particularly if you're trying to decide whether these people require additional doses, for example, of vaccination, or how good is their protection? How is their immune status evolved in comparison to the pandemic? And this was a cohort that we've followed and are continuing to follow now of dialysis patients in Germany. And comparing them, in this case, to control individuals who were actually doctors of theirs from the same clinic. So we knew they had roughly the same environmental exposure. And we could see straight away that in both plasma and saliva, there were significantly impaired responses within this dialysis cohort. The individuals that you can see in yellow are people who were also taking additional immunosuppressive medication. So we could look at these and pick out also how you can see for some of them, it does make a significant difference. They drop down to the very bottom of the assay, but some people were still very highly responding despite taking these medications. And as we followed them over time, we could see that really actually they underwent horrifically bad drops in their circulating antibody levels post-vaccination. So this T1 was 3 weeks after they had received their second dose, and this T2 was 4 months later. And we saw there that already a large number of them, particularly this line that you can see at the very bottom, had effectively seroreverted and had no circulating antibodies present. And more concerningly, and this work was then done in July last summer, we could see that actually for the healthy controls, they were undergoing themselves around a 3-fold decrease in their antibody titer. So this was already giving us a sign that actually, while it was working and you were producing these antibodies post-vaccination, you were likely going to need additional doses in order to stimulate performance further and to increase these titers. And when we've then followed them now for post third dose, this is shown here with this green syringe in the middle around day 225. You can see here that this increased their responses massively, much higher than it had done, say, post second dose originally. But that this pattern of drop-off then continued, and they then continued to decrease. And it was actually now with this fourth dose, shown in blue syringe around day 340 now, that we are seeing they are generating even higher titers following now this fourth vaccination dose, but the individual nature becomes even more apparent here. You can see that you have large differences, sort of almost 50-fold differences between those highest responding to vaccination and those lowest responding to vaccination. And this is very critical then if you're obviously trying to determine vaccination strategies and identify those of your patients who are likely most at risk from it. And to really hammer home kind of this message and why then this sort of having multiple analytes is important, well, all of the data I've shown you to this point on this cohort was just for the wild type B1 isolate of the RBD. But when we now look at the situation was with the BA1, we see that while we had this very nice uniform to a degree or very high responding group after the fourth dose for wild type, as soon as you look at the BA1 variant, you drop back into the middle. You've had almost a 2-fold reduction again of their actual inhibiting responses against this one. And this work is now even, despite being pretty much impressed now, it's even further out of date because this only went up to the BA1 variant. We since had 2 different waves following it. So we can see that actually through assessing these differences in the antibody responses, we gain a picture not only of just their individual responses and how it's evolving in response through the pandemic with more doses, but also how their response is changing in the context of the pandemic and how it's changing in response to the different variants. So this brings me on to now some of the other pathogens that we work in within our lab. And one of these is Lyme borreliosis, or more commonly known as Lyme disease. And this is the most common tick-borne infectious disease within the Northern Hemisphere. It is caused by Borrelia bacteria, and ticks themselves act as vectors, so the actual transmission occurs through tick bites. And it is estimated approximately 5% of those who are bitten with an infected tick will become infected. However, we have no real understanding of what the actual prevalence is of this disease. And this is because it's very difficult to diagnose in part, but it's also very difficult to actually really assess how many people seek treatment for this. Not all cases are symptomatic. You do have a large number of asymptomatics. And so we estimate currently, or it is estimated that there are around 300,000 new infections per year in Germany. This was based on receipts given to insurance companies. However, this has a very large margin of error, and we could be looking at 1/10 of this, and we could be looking at 10 times this amount. And we're now seeing also with borreliosis, it's not just restricted to environments that it's previously been restricted. And obviously as a result of climate change and how we've also seen new pathogens start to emerge in previously colder environments, we are also now starting to see Lyme disease, for example, become more prevalent in the UK, where it had previously not actually been reported for a large number of years. And as I said, diagnosis of this disease is ideally based on clinical symptoms if you have this very nice bullseye rash, which is known as an EM. However, this is not present in all cases, and this also tends to disappear around 4 to 6 weeks after the initial bite. The majority of people, though, will experience very unspecific symptoms, similar that you would for a natural cold or a flu or something like that. And so in that case, serological diagnosis and antibody testing is necessary and is the current gold standard used within Europe. And for this purpose, currently there is initially an ELISA screening to test with very high sensitivity. And then this is followed up with an immunoblot for high specificity to identify people who have antibodies. However, this is not always possible, and you can miss several cases as a result of low seroconversion. This also requires very large amounts of material due to its current format of having 2 separate assays that require it. And this also means it's very time intensive within a lab as well. So what we have developed for this purpose instead is to use our multiplex approach of different analytes and also combine this with dual antibody detection. So we look at both IgM and IgG simultaneously. And by combining these 2 together, we can achieve sensitivity and specificity of around 93 and 96% in identifying cases of Borrelia. And this, particularly with its bead-based format and the fact that we can, as with all of our other assays, automate it, makes it really ideal for use in large epidemiological cohorts. So on the right here, this is some very basic data that was really just looking at different sites across Germany and assessing differences in seroprevalence. And I mean, I'm not going to go into this in detail, but this kind of work is only really possible by using these kind of multiplex automated formats, because if you did want to do this, for example, with the classic ELISA and immunoblot, you would need not only much larger amounts of material, but this would also take a much longer period of time to physically complete the screening. As I said, we have also other pathogens of interest within our group. And 2 of these respiratory pathogens, RSV and influenza, are now currently ones for which we are undergoing developmental work. So RSV are very common respiratory viruses. Pretty much everyone is infected with them at least once in your lifetime, normally in the early years of life. And if you are healthy, you will just have mild cold-like symptoms. However, if you are in a certain group, for example, children under the age of 3, older adults who have weakened immune systems, people who take immunosuppressive medications, then infection can be serious. And this is referenced by that within children under the age of 1, this is the most common cause of both bronchiolitis and pneumonia. Now, this is also a pathogen that has had a severe change inside the last 2 or 3 years as a result of the pandemic. So these sort of preventative measures that we have taken place, such as school closures, wearing masks, reducing this, increasing social distancing, mean that in 2021, the classic autumn and winter pandemics, when you would normally see the majority of infections, disappeared. And instead, we actually saw these seasonal epidemics completely change to the summer. So in fact, in Germany, it was from around May until August this year that we actually underwent the RSV epidemic, as opposed to traditionally sort of between December and March. And this absence of the circulation and the fact that people are not becoming exposed to this pathogen, this could have severe impact on certain groups. For example, pregnant women, as when the children are born shortly afterwards, there's going to be minimal antibodies that can be transmitted maternally through breast milk, but also these children will have not had the experience basically of coming into these kind of usual seasonal pandemics, and they will be coming in at a different time point where we are uncertain how it will respond. So we have begun to develop an assay that will really actually look at identifying RSV accurately. And for this, we use multiple different analytes, not only from different targets, such as the fusion protein or the G protein or the N, the nucleoprotein, but also within different strains themselves. And I will show very briefly the data that we have so far. So what we have identified is that when we can look at different analytes here, for example, the N and the F, we can identify differences quite nicely. So with longitudinal samples, we can identify whether there has been a new infection, for example, shown here on the F by this increasing sample from May 2020 to March 2021. And we also see that we can identify real general decreases over the course of time. So there's kind of rate of seroreversion that people undergo. And the key part is that everything that we have done so far, we have obviously kept on validating our approaches as we have gone. And we have seen that this is a highly reproducible assay. So this is something that we are actually now further expanding, and we are looking at really using these G proteins to see if we can differentiate between subtypes of RSV, so between RSV A and B, but also between potentially different strains of RSV and gain an idea of which strains are actually circulating currently. Influenza, the other viral pathogen that we are kind of looking at and really developing at the moment. Obviously, I think everyone is aware that this tends to be a bit more severe, particularly the high end. So while there are a large number of very mild cases, there are often very severe cases. And we have yearly booster vaccinations available against the circulating strains ahead of the flu seasons. While there are actually 4 types of it, it's A and B that cause the vast majority of infections, and these are the 2 that we have focused on. And there are, to give an idea of the diversity of influenza, there are absolutely thousands of different subtypes of it. So differentiation is made on the basis of this H hemagglutinin protein and this N neuraminidase proteins. And you will see with these ones, you have huge differences, and this is perhaps best to think of influenza almost like separate pathogens as opposed to strains within a single pathogen. And historically, influenza has also been, by and large, the cause of a large number of pandemics that we have had globally. So for the H1N1, we have had the Spanish flu back in 1918, post-First World War, and then also the 2009 swine flu outbreak. Within other types, we've had H2N2, which spread in the 1950s in China, and then the H3N2 in the 1960s from Hong Kong. And H1N1 is now one that is actually causing particular concern within the people who monitor these types of diseases, because we have seen that in the last couple of, particularly even in the last 6 months to 1 year, there have been unprecedented numbers of cases of it. So originally back at the end of last year, it was reported in the UK. It was spreading through wild bird populations, but at the time it was restricted to largely continental Europe. And now in this article from actually only about 3 weeks ago, we see that it has already spread to North America. It is spread through multiple species of wild birds. And obviously the more species that is spreading through, the more contacts and the more chances it will have to come in contact with people and potentially switch and start infecting humans, and then we're on the process of having another pandemic again. And so we have been really looking at now developing these assays, and particularly for us, a key focus here is where possible to actually try and get differentiation on the basis of serology between these different subtypes. So what we have seen in our very early work is that particularly among individuals who are very young, who have likely only been exposed to 1 or 2 potential pathogens, we can use these G proteins and these H proteins to differentiate between people who have had H1N1 infections versus H3N2 or H7N9 or influenza B. And so here, this is just an example of some individuals that we could find who have only been exposed so far and only been infected with H1N1 or with H3N2. And obviously this work is now continually ongoing. We will have to obviously factor in vaccinations and see how this changes the story and how these assays can work. But this has already given us good promising results that at least this type of differentiation by serology and through multiple analytes should be possible for this pathogen. Lastly, I'm just going to talk about emerging pathogens and the work that we're doing now on monkeypox. So I think this is perhaps the most up-to-date work that we have within this presentation. And obviously this year, we have now had the first time that we have had large spread of this pathogen and this disease outside of traditional areas such as Central and West Africa. So going back to sort of springtime earlier this year, we had around 57 cases in the UK. I think in Germany, we were at similar kind of numbers around the 50 to 100 mark. And obviously this has now escalated substantially, leading to in July, the WHO declaring this a public health emergency of international concern. And the last time a pathogen was declared this was SARS-CoV-2. Now, unlike SARS-CoV-2, we already know that we will have antibodies present within the population. So there has been historic smallpox vaccination. The exact time point at which this finished will obviously depend on the country itself and its public health strategy. But there have also been a newer developed smallpox and monkeypox vaccine using vaccinia-based technology. For monkeypox itself, one of the real tricky parts of really developing any assay is there is very high cross-reactivity between the orthopox viruses to which it belongs to. So this is not just monkeypox, you also have variola, smallpox as it's more commonly known, as well as typical rural pathogens such as cowpox. And monkeypox itself, this diagnosis is obviously normally based on PCR of skin lesions plus taking patient history. So we are not advocating in any way for this using serology to identify cases. Instead, we are actually envisaging that serology here could be used to identify on a population-based level how many people have antibodies still circulating, particularly those who received this historic smallpox vaccination or this new vaccinia-based one, and really to identify and understand if the situation does escalate. I believe now in Germany we are around 3,500 to 4,000 cases. If this does grow, how much of the population already has antibodies that could bind against this? And more critically, are there any groups that we can identify that are not having antibodies present against this? So to begin with, we have really started just working on testing out various different antigens. So you can see here, we can actually, using our system, screen large numbers simultaneously, and we could identify fairly easily and very early on that some antigens had much higher value for when it came to really identifying antibodies produced through either infection or historic vaccination compared to just the standard population. So you'll see here, the A44R, we do not really see any differences to, and this is not an issue with the antigen quality itself. This is because the A44R has around 98% sequence similarity to these cowpox, to goatpox, to smallpox, and things like that. And so the cross-reactivity is too high to be overcome with this antigen. But by comparison, if you look at B2R, you'll see here that actually you get very nice separation from both the infected and vaccinated individuals compared to negative. And so while there is still obviously this large variation, you can see how large the whiskers are on these box plots. It at least is showing us already that we actually have certain antigens that we can use to really differentiate and pick out people who have already some kind of antibodies present towards this pathogen. And that this pattern is not consistent not only for IgG, but we can also look at this for IgA. And here we can even pick this out further and really separate even more the differences, for example, between people who have been infected and people who are vaccinated, who obviously have minimal to no circulating IgA present. And I would end this part here by really just saying that we obviously, as I've stressed throughout this, continually validate our assays as we go. And we could see here that even for this new pathogen and for these new antigens, at every step of the way, we can confirm the assay is stable in performance, we can confirm that the antigens are stable, and we can see that what we are building is already conforming to the analytical standards to which we hold ourselves. And so with this, I would bring in a summary. I hope I have been able to show to you today some of the uses of multiplex serology and the insights that we can gain from really differentiating out these antigen-specific antibodies. I think I hopefully have also been able to show you that our modular-based format that we use enables this quick development of new assays and integrating new assays into response to changing pathogens or the development of new pathogens, and that these information-rich assays, they are of interest not just for academic partners, also for industrial partners. And with that, I would thank all of the individuals listed on the screen. There are many more who I should thank, but particularly Thomas Ewers and Nicole Schneider-Hanmara, who were the former head and current head of the biochemistry and multiplex immunoassay groups. Large amounts of this work that I have shown here has really been the work of 4 of our PhD students, Matthias Becker, Daniel Juncker, Patrick Marzal, and Julia Herring, who have really pioneered large amounts of this assay development. I would also thank our similar groups here within the NMI of the Biological Development Center and our Bioanalytics group, plus the various funding organizations and partners for samples that you can see here indicated on the right. And with that, I thank all of you for attending.
Okay, thank you. Thank you very much, Dr. Dulovic. It's a very fascinating talk with tons of data. So now I think we can jump into the Q&A session. So I can see already there are a number of questions. So the first one from Yu Qi, and he asked what criteria, for example, with tags, do we require on antigens in order to get them to work in our assay format?
So we are fairly flexible with this. Obviously, classic his tags and stuff are preferential. Really, this is for us not a particular issue. We see that we are not restricted in terms of the type of coupling that we use. So we don't just use, for example, classic EDC-based coupling. We also use a more modern technique known as Anteo, after the company that developed it. And for this, we see actually we have done work where we've also used, for example, beta-alanine tags on peptides. We are incredibly flexible for it. And really, it's actually a case that we can speak to the manufacturer and find out from them what is the most optimal tag for them to use to really purify this antigen and produce the highest quality protein for us. And we can then flexibly fit this with our beads.
Okay, thank you. And from the same person, so in terms of the sensitivity, at which range of antibody concentration can be detected in the methods?
So this is very analyte specific. So the types of assays that I've shown by and large here, these are not fully quantitative ones. So we don't use standard curves to determine this. However, for similar work that we have done, we see reliably we can go down to the picogram per milliliter range.
Okay, and as a following up question, is there any difference between the IgG and IgM format antibody detection?
This, I think, by and large, we see with the type of assays that we use that the IgG tends to be a slightly more stable response than the IgM. I don't think we've investigated explicitly if there is a difference in sensitivity between the two. This will be an interesting point for us to look at. But I would expect probably the IgM, while there would be a small reduction, I think it would still be fairly close.
Okay, great, thanks. And the next one from Florian Huber. Which expressing system host organism do you use to express antigens, and how do you ensure correct folding post-processing of the antigens?
So expression system, again, we are flexible. I think the majority that I have shown here were based on HEC293, but we also have antigens that were expressed in E. coli, as well as those from baculovirus. In terms of how do we correct, I think it was correct folding and post-processing. So there is the standard analysis that is done by manufacturers such as Sino with blotting and things like that, and confirming that the size is correct. We also internally always also assess the antigens that we receive through mass spectrometry. And this allows us to basically confirm that not only the sequence and size is correct, but effectively the protein is produced as we would expect it. For folding, we have also tested, for example, with SARS-CoV-2, using nanotemper and things like that to ensure that we have exactly the type and the structure of the protein that we are expecting.
Okay, great. Thanks for the information. And the next question is also from Yuqi, asking for the antigens that you conjugate on the beads. Do you prefer to use a full-length protein or the truncated protein? Because using yeah,
so obviously you will have differences in the epitope and likely responses from this. We would prefer to use the full-length protein. Obviously, some proteins this is not possible, and you do have to use truncated versions. But this is also nice of where we can use this multiplex format, that we are not limited in the sense of only being able to use one and only being able to test one epitope. We can actually assess multiple different ones simultaneously.
Okay, great. Thanks. And the next is from Team Kiatis. Do you develop any multiplex assays outside of Luminex platform? For example, CARA-based, what are the limitations of the Luminex system?
So currently we do not really develop outside of the Luminex-based platform. That is our kind of modular technology that we use for all of our development steps. In terms of limitations of the system, obviously there are more sensitive methods that you can use. For example, Simoa, if you want really sort of ultra sensitive measurements of certain analytes and stuff. It really is the limitations. I think it's best to describe them. The limitations are no different for any other technique that you use. It's you have to find the right technique that fits for the type of assay and the type of information that you want. For our assays, because we are obviously far more interested in not only having these kind of automated formats that mean we can use them in epidemiological cohorts, which also means we are willing to sacrifice a small amount of, say, sensitivity and specificity by doing it this way. But this works for us because for us, the key part is the combination of the number of antigens and the ability to screen a large number of samples simultaneously. So we are not so focused on necessarily having a super highly sensitive system that would allow us, would give us the most accurate sort of qualitative or quantitative answer. For us, our focus is different. So for us, the Luminex technology is perfectly set up for this.
Okay, so so next one is from Nathaniel asking, do you observe differences in background signal when working with antigens from different cells?
We do observe differences in background signals between antigens, between different cells. This is tricky to say. It's very rare that we can source an antigen that's been produced in multiple different types. It tends to be that if its antigen is made in hex cells, everyone makes it in hex cells. If it's made in insect, everyone makes it in insect, for example. I can't really answer that question for differences between them. I think we obviously see batch specific differences, so there are small differences between them. However, largely background signals are fairly stable within the format we use. They are, for example, always under 100 MFI by and large. And I think if you can sort of cast your mind back to most of the slides I showed you, the actual antibodies of interest, these are measured in the thousands instead. So the background for us is not too much of an issue.
Okay, and I think that's all for the questions. So if I may ask, I also have one maybe still bit question. So do you see differences in assay performance between the automated measurements and the manual measurements?
Yeah, you will see small differences between the two. This is because, simply put, the robots, they can't sense if a sample is shall we say, not optimally prepared. So if it's hemolyzed or something like that, or if it's got a contaminant of some description, whereas a person who was working under the hood can see this, can adjust for this, particularly if there is some kind of large visible contaminant like fat droplets, you can pipette around this. So you will see that particularly with certain sample materials like saliva, you do undergo reductions in your sensitivity and specificity by using automated techniques. However, the trade-off of this is that one person who is working full out can realistically get through maybe at most 1 3/8/4 plate in a day, whereas with an automated technique, you can do 2 comfortably, 3 fairly easily, 4 if you do shift work. So this offset of the increased number of samples for our purpose makes up for this loss of sensitivity that we see.
Yeah, understand. Thank you. So I think that's all for the questions. So if no more questions popping up. Okay, so I think that's it. Thank you very much, Dr. Dulovic. Thank you very much for your time. It was really a fascinating talk. We really appreciate that you're spending a lot of time doing it. And before everybody departs, so I just want to mention that at Xenobiological, we have the world's largest collection of recombinant proteins with a very broad range of viral antigens, cytokines, growth factors, and also biomarkers. And we also have more than 14,000 different antibodies for you to choose. So you are always welcome to contact us whenever you have a demand for our recombinant proteins, antibodies, or for related customized research service for recombinant protein expressing or antibody development. So with that, I now want to thank all of the attendees who joined us today. I wish you all the best in the future. And thank you very much for today. Thank you very much for your time today. So we will see you in our next webinar. Have a good day. Bye. Bye-bye.